SDS PAGE is a technique used within biochemistry labs to isolate proteins and fragments based on their molecular weight. So SDS PAGE, it stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Now this sounds like a big mouthful, but if we break down each of these terms, we can figure out what is happening. So sodium dodecyl sulfate is essentially the name of the molecule that is going to bind to our proteins and carry it through the gel, okay? So sodium dodecyl sulfate is just the molecule doing the carrying. Then we have polyacrylamide gel. That's just the name of our gel within our apparatus. It is a cross-linked polyacrylamide gel, and we can alter the percentage of this gel, which will affect the pores within the gel. So it's going to affect how far our proteins or fragments travel. Now, let's say we run SDS PAGE, we have this negative end, we have this positive end, so our negatively charged sodium dodecyl sulfate is going to move towards the positive end. And the smaller molecules, they will be able to travel further because small fragments have a smaller molecular weight and they are able to travel further, whereas our large fragments, or the ones that have a very high molecular weight, they will be near the top. So essentially, this molecule has a negative charge, it's moving towards the positive end, it binds to our protein and it carries it through this electric potential where our small fragments will travel further. Now, once we run SDS PAGE and we take out this plate over here, we won't be able to see how far our proteins have traveled. We would have to stain the plate first, and we stain it with Kumasi blue. Once we stain it with Kumasi blue, we get this more visual representation of how far our fragments have traveled. So in our first column, we always have our markers, and our markers will be our known values. So it will give us our known weights. And we compare the markers with what we applied to each of these wells. Now, if we take a look over here, we can see that in our cation exchange and anion exchange, we tried to isolate a protein and we run it through SDS PAGE now, and we get this band over here for anion exchange around 45,000, and for the cation exchange, we also get a band at around 45,000. Now, what does this tell us? What can we determine from SDS PAGE? So the number one thing that we can determine with SDS PAGE is the purity of our sample or contaminants that exist within our sample. So let's say we did an anion exchange column to isolate a specific protein. Once we've isolated that protein, we run it through SDS PAGE, and then if we get multiple bands, that means there are other things, other contaminants within our sample that are giving us these various bands. But if we only see a single band, that means we only have one type of fragment in there, which indicates that our sample is pure. So in this case, in our anion exchange column, we see 1 mark. Cation exchange, we see 1 mark. So whatever protein or fragment we had, it purely exists alone within our sample. So we would say it's a pure sample. Now, if we look at this column over here, we can see that there are a lot of contaminants. We have various bands at various sizes. Now, another thing I want to make note of is that we have these lighter bands and we have these darker, thick bands. The lighter bands just indicate there is a small amount of whatever it is. So here we can see a little bit of contaminant over here, right above 45,000. Below 45,000. And we see this thicker mark over here, which means this predominates more in our sample, and we have a little bit of contaminant over here. And just to make note of the markers or standards on the side, we can see from our values that here we're starting off at 97,000 and we're moving towards 14,000. So this just shows us that our smaller fragments, they are traveling further, and our larger fragments with the high molecular weight, they're the ones that are traveling slowly, so they're going to be left behind up here. So when we look at SDS page, we look at our stained slide. The smaller fragments, they will be closer to the bottom, whereas our larger fragments with the high molecular weight, they are going to be at the top. So SDS page, it is based on molecular weight, where sodium dodecyl sulfate is the molecule that is carrying our proteins or fragments through the gel. And that is essentially SDS page.